Hi, welcome to AP Biology. Today I want to go over facilitated diffusion. So let's put it in context of where we are in the notes. So we're looking at how things move in and out of cells. And so we have passive transport versus active transport. Passive transport requires no energy input, as opposed to active transport that does require energy input. That energy input is usually in the form of ATP, but not always. But passive transport doesn't require that, and that's where we are today. So facilitated diffusion is the movement of materials in and out of a cell membrane through a helper. Facilitator means a helper. So let's kind of just talk about these other types. Simple diffusion, osmosis, and facilitated diffusion. These are all types of diffusion, which means materials are moving. So materials move from a high concentration, this symbol means high, to a low concentration. So that means down a concentration gradient, or with a concentration gradient. So that can mean, let's say you have a cell here. Let's say there's a lot of oxygen outside the cell. Let's say these little dots are oxygen and not too many of them inside. And let's say you've got lots of carbon dioxide in the cell. And so these blue dots are carbon dioxide. You have a little bit outside, but mostly inside. The concentration here from high to low would be going out for carbon dioxide, but for oxygen, it would be going in. So you can have concentration gradients in opposite directions, but if it's passive transport, it's always going to be going high to low. That might mean into the cell or it might mean out of the cell, depending on the particular concentration gradient. So simple diffusion is the movement of materials right through a lipid bilayer. So the kinds of materials that can go right through a lipid bilayer are nonpolar molecules or lipids, or small nonpolar molecules. So oxygen and CO2 go right by simple diffusion. But let's say there's a material like sodium ions or sugar, something bigger, polar, something charged. That can't go right through the lipid bilayer, so it's going to have to go by facilitated diffusion instead. And osmosis is the movement of water, could be either by simple diffusion or could be by facilitated diffusion. So let's talk a little bit more about facilitated diffusion. So if a cell needs a material, like maybe glucose or maybe sodium, and it can't go through the lipid bilayer because it's charged or it's big and polar, how does it get into or out of the cell? Well, it could go through a protein. Like here, that's a channel protein. So a channel protein is sort of like a tube or pore. Or it could go through this carrier protein. A carrier will change shape, so we'll talk about those a little bit more later. So it can go through a protein or it can go in and out through a vesicle. So either a vesicle is pinched off going in or a vesicle transports stuff to the surface. So we'll talk about that in a different video, but we'll talk about the proteins today a little bit. So facilitated diffusion. Transport proteins allow polar or charged materials to move through the membrane. So the diffusion part means that materials go high to low concentration. So you have lots of it right here, and you have very few of them here. So you're going from where there's lots of them to where there's very few of them. That's sort of the natural way to go, high to low. Entropy, things sort of spread out. That's all. The facilitated part means that there's a protein that helps it because it can't just go through the lipid bilayer. That means you can control it. So you can have a cap on this that closes it off and says, you know what? I know you want to go high to low, like that's the natural way for materials to move, but I'm going to close you off and not let you go right now. So if the materials are moving from high to low concentration, then it's diffusion. This does not require energy, so no ATP input for this. The proteins are very, very specific. So this protein channel, maybe it's only allowing glucose through. It's not going to let any other material through. They're very, very specific, which means you can control them. You can decide whether you want glucose to move through the cell membrane and how much of it, because you can close it off a lot of times. So here's a picture that I like, but I don't know that it makes a whole lot of sense. So this pink thing here is really it's really a protein that like the whole back and front has been cut off. So imagine it looks more like this, right? It's sort of like a circle. And materials are going from where there's a lot of them Let's change color here. From where there's a lot of them, here you have a high concentration, and they're going right through this tube to where there's a low concentration. And you just see like the edges of it. You don't see the back and the front of it, which I think is a little bit weird. It's sort of more like this. It's this little tube. So types of transport proteins. These channel or pore proteins are really just a tube. And this one's been cut like both the back off of it and the front off of it. Anyway, they have hydrophilic channels that certain molecules or ions can use. So in the inside, it'll be hydrophilic. Maybe out here, you know how there's water outside of the cell and water inside the cell. So out here, the R groups on the amino acids, because they're near water, out here the R groups would probably be hydrophilic. But, and maybe in here along the part that's near whatever it is that's moving through, maybe sodium's moving through and you might have some like negative, partially negatively charged R groups out here, right, to attract the sodium and help it to go through. Out here though, right on this part, do you see how this part of the protein is near the lipid bilayer, right? This thing is the lipid bilayer, right? And so right here along the whole outer edges of that protein, you might have hydrophobic R groups in the outer part of that protein. So AP does want you to know when proteins fold, the hydrophilic parts would be where they should be, out near the water. Right here, you'd probably have hydrophilic R groups. But over here, the parts of the protein that are snuggled up against the lipid bilayer out here along the whole outer edge, meaning the part that's in the middle of the lipid bilayer, that would be all hydrophobic R groups. Okay, carrier proteins can change shape to move stuff from one side of the membrane to another. So maybe there's like a little hydrophilic part here that then closes off when it goes back together, right? So it can move through the lipids. But anyway, a carrier protein is going to change shape. If this is facilitated diffusion, it's still moving stuff from a high concentration to a low concentration. It still doesn't take energy to do this. The protein just has different states that it can be in. So if this is a high concentration of little red dots out here and you have fewer of them here, then mainly those red dots are going from high concentration to low. But it's sort of random. Like if you ended up at some point having more of them here, it just as easily would bring them back in the other direction. Not using any energy for it. Here's another carrier protein that just spans the whole cell membrane. So I just wanted to show you that example. In this case, the high concentration is inside, sent out to a low concentration outside. Okay, next idea is that osmosis. Remember, osmosis is the movement of water. So that can be simple diffusion because even though water is polar, there's so much of it and it's so little, it can actually go through that lipid bilayer. That's the lousiest picture I've ever drawn of a lipid bilayer. Anyway, it can go through the lipid bilayer, but osmosis can also move by facilitated diffusion. So if the water, if you have these things called aquaporins, these are proteins, and so this is like the loop, looks like kind of an alpha helix here, loop to loop to loop, right? And right here, this section through, I guess the lipid bilayer right here, this section through is where the aquaporin is, and the water here's outside extracellular, and this is going to be inside where the cytoplasm is. This is a tube that sort of separates out the lipids a little bit, these phospholipids. So you'd have some phospholipids here maybe, right? And this tube, this protein, is going to allow water to move through faster. So an aquaporin is a protein that allows the passage of water to move through the cell membrane at a faster rate. And so it's going to be like sort of a narrow thing that only lets the size of a water molecule through. And this would have a positive charge right here in this R group that's attracted to the negative part of water and just helps it to move through.